Hi, I'm David Carlson. I'm an associate professor at Duke University. And today I'm going to be talking about machine learning to infer and control brain state and some of the things that we can do that are really fun with that. So, just to start this out, I want to start talking about what is a brain state. So, when we start talking about a brain state, we actually are thinking a lot about a subject, right? This could be, in a lot of the examples we're going to use here, an animal model, but this can also be humans. And what we're going to be able to do is we're going to take a lot of measurements about what's going on in our brain. And we're going to take all this data about what's going on in our brain. And we want to be able to extract it out and we're going to summarize it. So we're going to take a lot of brain data, use a lot of statistical techniques there to summarize all the information about what is currently going on as an internal state, what's going on in the brain. And we're going to say that this is going to be broadly useful to understanding the types of behaviors that might be happening. So, for example, if we're looking at a mouse, it might be in a happy mood, in a social mood. So we might be looking at this animal and we might see that it's in a happy social mood, it's engaging in prosocial behavior. And this is what the brain state is saying. It's saying that this is the type of behavior that we're going to engage in. And alternatively, it might actually say that, hey, we're actually going to be able to tell that we're about to engage in very aggressive behavior. Now, in the wild, sometimes aggressive behavior is very appropriate, but sometimes it's actually pathological. And this is where we start to have challenges. So one of the things that we wanted to be able to say is that if we can find this brain state and we can understand exactly what's going on in this brain state, we can actually start to do interventions. So what we're going to say is if it looks like we're heading down a path, we're going to have inappropriate aggression, let's make some sort of intervention. And I'll talk a little bit more about what this intervention is as we go through the talk. We'll make an intervention and we can actually try to restore normal behavior. We can actually change the brain state. And by changing the brain state, we're going to be able to change the type of behaviors that we engage in. Now, there's this really big question here of, okay, this seems a little bit complicated. How can we actually do this? Well, in my research, we are using machine learning or artificial intelligence, and we're going to be able to learn brain states, and we're going to try to use this to design interventions, right? So we're using a lot of machine learning methods to learn brain network models of emotions and behaviors, right? And so one of the big things here is that we want to be able to design interventions from this. So we need to be able to take these methods and we're going to need to be able to summarize the results or the type of insights that we make from it. And we need to be able to communicate with neuroscientists, right? And the key idea here is that if we can use explainable techniques, we can use explainable AI to generate testable hypotheses. And even going beyond the fact of testable, I actually have been working a lot to actually try to design confirmatory experiments or interventions. We can come up with theories of how the brain works. We can come up with theories of what's going on in the brain state. And using this, we can actually design confirmatory interventions by actually working together in order to try to do this in a collaborative manner. So a lot of the experiments that I'm talking about here are joint with a collective for psychiatric neuroengineering, which is led by Kafu Duraza and myself as well. And what we're going to be doing here is we're going to be looking at an animal model. And we're going to have brain implants. We have a lot of electrodes implanted in a number of different brain regions. And what we're going to do is we are going to extract out this brain information. We're going to take this brain information, and then by using a machine learning technique, we are going to find some sort of brain network here. I'm calling this an electrical functional connectome because it's defining what's happening in the functional connectivity patterns of these different brain regions. And we actually, from this, we are going to be able to communicate with a neuroscientist and use this to design or choose an intervention. And then from this, we'll actually manipulate the brain activity. So we can do this in a full circle where we can actually use the AI ML to understand and summarize what's going on in the brain. And then we can use it to actually design experiments that we can use to test these hypotheses and directly manipulate the brain activity. So to explain how this works, in the framework that we're using, we're going to be using local field potentials. So these are depth electrodes implanted in a number of different brain regions. Local field potentials are often thought of as just like the brainwave. So if you think about what happens in brainwaves. You have your standard different brainwaves, like what happens in alpha, beta, gamma, etc. And we are going to get this from depth of electrode in many different brain regions. And we can think about what happens in this in different wavelengths or different bands, and we can think about how much power there is in each of these brain regions at different frequencies. But we can also think about, well, what is the type of information and what can we capture about what's being shared between brain regions? So we're also going to look at connectivity patterns between these two different brain regions. And in particular, we're going to look at the connectivity patterns between these two different brain regions at a particular frequency. So what I'm showing you here is I'm showing you a brain pattern at a particular frequency in the hippocampus and the prefrontal cortex. And what you can see here is that a lot of the peaks are matching up. This is a highly coherent signal. And we're going to use some Granger causality to actually try to capture directionality here, because we want to be able to know not just are these brain regions communicating to one another, which would drive coherence, but also are they actually sending information to each other, right? And one way we can do that is by actually saying, do we actually see the signal of the hippocampus come through to the prefrontal cortex? If we see this kind of lag here, then we can say that information is being sent from the hippocampus to the prefrontal cortex. And there is, of course, a mathematical definition of this, but I'm going to skip past that. And likewise, if we see this other relationship where the peaks in the prefrontal cortex tend to lead the peaks in the hippocampus, we can actually say that there's a statistical relationship where the prefrontal cortex is sending information to the hippocampus in this particular frequency. And so when we do this, we want to describe our brain network or electome network in these types of patterns. So what I'm showing you here on the left in this wheel and spokes is that we have a number of different brain regions here. And within each of these brain regions, so for example, if we look at the top left of this plot on the left, it's the ventral tegmental area. We have down beneath it 10, 20, 30, 40, 50. This is the frequency that we're looking at. And on the outside, we can see a band if there's a higher amount of power during this brain network. And then we can also describe the connections. So if we have a connection between two brain regions in a particular frequency, this is just showing that we have some sort of communication, right? There's some sort of coherence between them. And the color here is not, doesn't have any intrinsic meaning. We're using it to identify that there are two prominent frequency bands that are identified here. So if we look on the right, one of the other things we can do is just say within this brain network, we're going to be able to look at, say, what is the information from 2 to 8 hertz and say, where does the signal originate and where does it go to? And so we can look at this and say that the nucleus accumbens is where a lot of this information is originating, and it's sending a lot of signals to the rest of the brain. Likewise, there's a lot of different patterns and different frequencies that are sending information. So we have a lot of brain regions that are the source of information, a lot of them that are receiving the information. Now, an electome network is probably complicated enough already because there's a lot of things going on. But one of the things that happens is that our brain does many things at once, right? So right now I'm giving a presentation, and within this presentation, I have been thinking about what's going on in my next slide. I'm in a pretty good mood. It's fun to talk about science. But generally, we feel many things at once. Our brain does many things at the same time. And so we need to have many brain networks that are being active simultaneously, right? So we need to have some sort of way of thinking about what is happening in all these different conditions. How do we have these two different brain networks? So when we start to talk about our model here, we are going to be using a superposition model of these brain networks. So what I'm showing you on the right is what the superposition model looks like. So at the very far right, where it's in green in the electome networks, where we have F1, F2, F3, these are 3 different brain networks. These brain networks are constant through time. And what we are saying is that when we look at what's happening at the current point in time, which is marked by W here for window, the current point in time, you have a superposition of all of these different electomes, and they're going to be weighted differently, or they're going to have what we can call a different expression. So we can say that the weight of this first brain network at the current point of the time is for example, SW1 there. And that can either be very high or it can be very low, and it can vary over time. So the brain networks are constant in this model, but this network expression is allowed to vary over time. So for example, if you're becoming happier and happier, that brain network related to happiness can go up and up and up and up. Or if you have sad or anxiety about presentation, you can have different sorts of patterns that are being expressed. And so the idea is that if we can find these brain networks, we'll be able to actually look at the type of intervention that we want to do, because some of these brain networks will be higher than others, right? So if we can find things related to aggressive behavior that's either higher or lower during aggression, we can find a fundamental network that is related to aggression. And so when we're trying to do this, it's actually very important that we think about all of these brain networks, that just learning them directly as a superposition model is a very nice framework, because it allows us to capture the heterogeneity of what the brain is doing at any given point in time. But one of the things that we want is that we want to be able to operate on a single brain network, if at all possible, right? So when we're thinking about aggression, it's much nicer to find one network related to aggression. And so we put an additional constraint into the model where we have these unsupervised part of the model, which I show on the left, which is exactly that brain superposition model that I showed you before. We have our electome networks, and then we have our network expression, and that is combined to make up our observed local field potential at this current time. But the other thing we want to be able to do is we want to be able to link our network expression to behavior. This matters for a couple of reasons. One is that we really want to be able to predict behavior from this network expression. So if we have some sort of behavioral label or behavioral information, we want to be able to guide the network expression towards that. But the other thing that we want to be able to do is we actually want to be able to say, hey, this network expression only at our first electome network is related to behavior. And if we limit which ones we are training or encouraging to be related to behavior, it allows us to put most of the information in a single brain network. So rather than saying defining 20 brain networks, which might be what's necessary to describe the full heterogeneity of brain activity, we can try to find and force the information related to one behavior or one relevant set of behaviors into a single brain network, which will allow us to look, rather than at the full set of 20 brain networks when we're doing our analysis, at a single brain network, or perhaps 2 or 3. And just as a point, what we're going to do is when we're thinking about building the system, we actually want to be able to take all of this information, and we want to be able to do this fast, and we want to be able to do this in real time. So we've set this up so that we can approximate this network expression with a neural network really, really fastly. So we can generate the features pretty quickly out of this local field potential. We pass it directly into our neural network, which can give us our network expression in the matter of a few hundred milliseconds, which we're talking about local field potentials here, typically a window size of 1 second. So 1 to 200 milliseconds of lag is a very appropriate measure. So we can do this pretty much almost as fast as what we're getting the data as. So this is the general model, and I want to talk about how we can use it. So we're going to start talking about what happens in aggression. So we're going to be looking at 3 scenarios where we have a male mouse that we're recording data from, and we're going to pair it either with another male or another castrated male or a female mice. And in these 3 different scenarios, you would expect different types of behaviors to happen. So there's a lot of different social behaviors that can happen here. So for example, we can think about attack behaviors such as biting, boxing, tail rattling, or tussling, which is what we're going to describe as example behaviors that we're going to classify as attack. We can also think about non-attack behaviors such as sniffing and grooming and resting. And these are not kind of antisocial behaviors. These are all social behaviors because we're all engaging with another mouse, but we're going to break this down into attack behavior and non-attack behavior. And what we're going to do is we're going to record this from a number of different mice, and we're going to apply that model that I just talked about to try to learn a brain network that is related to attack behavior and can help us identify when we're going to engage in attack behavior versus non-attack behavior. So the network we found here is on the right. And I don't want to belabor the network too much, right? I want to just say that this is what we found. There's a lot of different brain regions involved. We can evaluate its predictability. It's predominantly driven by a couple of different bandwidths. So we're in particular overlapping with theta and beta oscillations as well, as long as some higher frequencies. And nearly every brain region that we measured was involved in this. And I will actually say, I'm not going to go over it, but we can actually show that including this many brain regions does include improved predictive performance over using one brain region or two brain regions. So there's this question of, well, okay, we've been able to derive a network using machine learning from this assay. And there's this really big fundamental question that we have is this real, right? I have gone through a procedure where there are some assumptions made about what's going on in the brain. There are assumptions about the model. There are assumptions about the structure of the brain networks. We're describing, in fact, a lot of functional relationships. And we want to actually say, well, is this network that we found, is this actually going to be something that if we manipulate or control, will it actually change the behavior that we want it to? And so we want to start thinking about different types of generalizability that we can think about. So when we start to say, well, we found a brain network, how well does it hold up, right? So the first level that we might think about is, what is a new subject? If we have a brain network, we get a new subject in, will it hold up in this new subject? We can also then start to think about, well, if we have a different type of assay or a different type of task, will we get the same type of response along this brain network, right? If we have other assays that we know relate to aggression, will it hold up in this case? And then if we actually can think about, if we do manipulations, does it actually respond in the way we want? Will it hold up in different genders or sexes? Will it hold up across different strains of mice? And then eventually, will this type of relationship hold up across species? Now, clearly, we're not going to get to every level of generalizability when we start, but we wanted to start looking at this. So there is this question of, does this generalize, this brain network that we found related to aggression? And the answer is yes. So I'm not going to show you the subject level data. It holds up very well across subjects. But there's a couple of things that I want to tell you. One thing is that, well, oftentimes we can discount local field potentials because the real currency of the brain, it's often believed, is neural spiking. And so what we can say is that a lot of the neural spiking activity that we observe is related to these brain networks that we're finding. So if we're looking at, for example, at this network related to aggression, 18% of all cells correlate above chance with the network expression related to this network. If we look at a different type of behavioral paradigm where we expose a mouse to a urine from a peer or somebody that they're competing with, we do actually see that it changes the brain network expression. So if we look at them where they have a clean room versus where they have a room where there is a smell of urine from a peer, we can actually show that this is related and changes the electome network inhibition, which is what is going to actually be related to aggression. So because this is an inhibitory network, as it goes down, you're more likely to be aggressive. So as a urine case puts you in a more aggressive case, this is exactly matching our hypotheses. Also, if we look at the type of designer drug where we can actually say, change what's going on here, we can show that we get a dramatic increase in the number of attacks when we use a designer drug that's known to increase aggression. And likewise, we again see a decrease in the activity level of this EN inhibition network. So we also want to be able to use optogenetics. So for this audience, I think people are probably pretty familiar with optogenetics, but briefly, this is a way that we can actually use light to make cells fire on demand within particular brain regions through a variety of techniques. Now, at first glance, it can be a little bit funny to think about brain networks that we're measuring from local field potentials through neurostimulation, where, especially in optogenetics, it's going to be acting on individual neurons. So if you look here, if we have some sort of therapeutics or intervention, this is going to actually manipulate neural activity. It's not going to directly manipulate the local field potentials. But by manipulating the neural activity, it's going to change and manipulate the population level activity. And from there, it's going to actually change the network level expression that we have in our electome models. And so we have this big full feedback loop where even though our electome model isn't directly acted upon by the therapeutics, we can still use it to design and get responses because we have this full cycle here. Where the intervention will act on neurons, which will give rise to the network level expression. Okay, so we first started out by looking at a neurostimulation which was known to try to decrease aggression. So what we've done here is we have done direct stimulation of the infralimbic cortex at 20 hertz. And what you can show is that if we look at a color that's not supposed to react to where the neurons are not supposed to react to the light, which is yellow, there should be no intervention, versus a blue light, which will actually manipulate these cells and make them fire. We can show that when we actually do this, we get a significant and very significant decrease in the amount of time spent attacking. So when you do this sort of intervention, you are seeing a lot of decrease in attack bouts and also in attack time. But one of the problems is that it also decreases prosocial behavior, right? So if we look at how much time is spent in prosocial behavior with a female mouse or a castrated mouse, it's also greatly decreasing those. So really what's happening with the stimulation is it appears that it's not just decreasing just aggressive behavior, but it's decreasing all social behaviors, right? It may not be aggressive anymore, but it's also not engaging in prosocial behaviors. Which goes back to where I started this talk, where we were saying that, well, okay, let's think about this case where if we have this brain state and we can actually understand what's going on in this brain state, well, if it looks like we're heading towards an aggressive scenario, we can actually have an intervention. And from this intervention, we can try to restore this to a more prosocial state. So rather than engaging in aggressive behavior, we'll have more prosocial behavior. And likewise, we don't need to stimulate all the time. We only need to stimulate in the cases where it looks like we're going to be engaging in inappropriate aggressive behavior. So if we look at this scenario where we are stimulating only when necessary, well, we can actually think about in this case where we have a bidirectional neural interface. We take our information out of the brain, we encode it pretty much in real time to a network level expression, and then we stimulate based on a certain threshold from our network expression, which will manipulate them on demand as necessary. So when we do this, the first thing we wanted to check is, well, we have a brain network that we are using to trigger the stimulation. If we look at the case where we're looking at the blue light versus yellow light, where the blue light is supposed to trigger it, if we look at this network, where lower means you're more likely to be aggressive, when it drops low, we stimulate. In the case where it's yellow, we're not supposed to have a response. Because it was so low, there is a little bit of a regression to a mean and it comes back up a little bit. But when we stimulate, when we have that blue light, we can see that our network level expression after stimulate shoots way up very dramatically. So the first thing we showed here is that this network really is responding. The other thing we wanted to look at is this actually good at controlling aggression? And the answer is yes. We see a very significant reduction in the amount of time spent in attack behaviors. But then when we looked at these other scenarios where we look at how they interacted with a castrated mouse or a female mouse, we don't have any significant effects on prosocial behaviors. So unlike before, where when we stimulated all the time, and you would see this great decrease in all social behaviors, here we're not seeing an effect on social behaviors in the castrated female case, and we are only seeing it in the highly aggressive case. And this is, again, stimulating only when we need to. And so there is this question of when we're looking at these types of controlled procedures, is it just something about the way we are doing the stimulation, right? Because we're stimulating a particular pattern now. Is this particular pattern just better at stopping aggressive behavior, or is it the amount of total stimulation? So what we did is we set up an example where we tried to examine this and control for this, where we looked at non-synchronous stimulation. So what we do is we take mouse 2, and rather than using its own brain data to trigger when to do the stimulation, we're using a peer mouse in a different session. And what we do is we use the pattern that should have been used for mouse 1 on mouse 2. And so in this sense, they're getting a very similar pattern to what this encoder would give us, but they're not using their own data to do it. This is a type of sham feedback, which is a very useful control case. And in this scenario, what happens is that when we look at, does this decrease aggression, which we would hope it would, right? If we think that this stimulation is what we need to do. When we look at it in this sham setup, we actually do not get a significant effect on male aggression going down. So it really does seem like the stimulation only if necessary is a really big advance. So the other thing that I wanted to talk about is the fact that, well, we actually have a lot of information in this brain network. So right now we're just using the brain network expression to actually analyze, does this work or not? Or when should we stimulate or not? And so what we can do instead is we can take this brain network and we can find very particular patterns that we think are going to control in this network. And what we can do is we can use that to design very particular neurostimulation procedures. So here we are looking at a very particular neurostimulation where we take a pair of brain regions and we only look along stimulation pathways of that pair of regions. And rather than stimulating this brain region broadly, we're only stimulating neurons in this brain region that end in another brain region. So this is a very particular pathway that was identified by this brain network. Now, if we think that this is highly related to this brain network, we think that this should actually lead to a lot of control for it. So even if we don't do the stimulation only when necessary, if we do this much more precise neurostimulation, we wanted to actually examine what does this do in our scenario. So when we look at what happens in the attack bouts, we get a very significant reduction in the amount of attack bouts. But when we look at non-attack behavior, it's actually very significant as well, but it doesn't decrease. It actually increases significantly. So when we look at this very specifically designed intervention that came from our machine learning model and the strong patterns it found, it greatly decreases attack behavior, but it actually significantly increased prosocial behaviors in one scenario, and in the female case had no significant effect. So just to conclude here, one of the really fun things is that we can actually identify a brain state and actively choose a manipulation in real time. I'm very excited about the power of using this type of framework to better understand the brain and also potentially move in the long run towards better interventions and treatments. And so we were able to find a brain network that encoded aggressive state, and we might be able to suppress aggressive behavior while not impacting prosocial behavior, which is something that I'm very excited about. We're currently working a lot on this, also in anxiety, depression, and in reward, to get a better understanding of what exactly is happening in all of these different scenarios. Just to acknowledge, we have received funding from the NIH previously. And this is a big joint effort, and there's a number of people here on the right who either contributed analysis techniques or experimental data. And just to conclude, finally, these are the references for anyone that would like to learn more. Thank you so much for your attention.